This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 43 for August 2nd, 2009. I'm Vincent Racaniello, and it's Sunday, and I was away again last week, so I didn't have a chance to record the usual This Week in Virology with other hosts. However, a few weeks ago, Dick Despommier and myself recorded part 2 of Virology 101, Virus Classification. So let's listen to that now, and I'll be back on the other side with some discussion of the 1976 swine flu vaccine and Guillain-Barré. Do you want to talk about virus classification?
Sure, because I would like to know whether or not they're classified based on their shapes, or their chemical characteristics, or the size of their genomes, or whether they're RNA or DNA viruses, or whether they're single or double-stranded, or whether... I would like to know all of that. So is that what you'd like to talk about next?
I'm happy to talk about that.
Okay.
How do you classify viruses? Actually, this is a very good point. You know why it's a great point? Because we probably throw virus names around and people don't know what we're talking about, right?
Exactly.
So, do I have a lecture on virus families? Let's see.
Why are they called families and not, the normal classification of life as Linnaeus originally conceived it and then was expanded upon by biology? We don't classify viruses this way because there are no species? Is that what you're saying?
So we use the classical Linnaean hierarchical system. What does that mean, Dick? You would know better than I.
Well, it's a binomial designation.
What does binomial mean? Two things?
There's a genus and a species name. A genus and
All
a
right,
species.
so there's a kingdom.
Okay.
Not for viruses, though.
No.
Do you know how many kingdoms there are? Plants, animals,
there are 5 altogether.
bacteria, archaea. What's the other?
5.
You don't know what they are, though.
There's prokaryotes, eukaryotes.
No. There's no more prokaryotes than eukaryotes. All right, let's.
There is a book called The 5 Kingdoms. It's written by Lynn Margulis, or Margulis as she pronounces it. And the 5 kingdoms are Protista and...
6 kingdoms.
6 kingdoms. We're up to 6
It's now been revised.
Okay.
You ready?
Yep.
Animals, plants, fungi, protists, archaea, and bacteria.
Right. What happened to the viruses?
Some textbooks only have 5. No, they don't have they're not living. They're not in the tree of life. But we are using a similar
another question.
So in the classical classification system, if you will, the Linnaean, you have kingdom, phylum, class, order. So we don't use any
family,
of those
genus,
for
species.
Yes. Then family, genus, species are what we use for viruses. So we start with families,
all right,
which are the words ending in AE.
Right.
Picornaviridae, Orthomyxoviridae. Okay? And when you'd use that family name, you italicize it. Or you can say coronavirus, in which case you don't. You just refer to it. So that's the family. Then we have genera.
Right.
Like enteroviruses, hepaciviruses within a family. Let's do a better job. We have enteroviruses, rhinoviruses, cardioviruses within the Picornaviridae. And then we have species within the genera. Polioviruses, type 1 is a species.
So is every virus particle a different species?
No.
I don't mean...
that's a good question because it's a quasi-species, as you know.
Right.
Many virologists don't buy the species concept. In fact, the plant virologists don't use any of this.
Right.
They don't believe in this classification.
I know,
they just give them virus names.
Right.
But the animal viruses, they tend to fall into family and genus. The species, a classical definition of a species are two organisms that don't reproduce, right?
Right. Or at least are genetically separate.
With viruses, it's
a messy
definition.
Yeah, it gets messy at some level because there are lots of exceptions to all the rules.
Well, that's why we make rules, right?
I think so. The moment you make one, they're broken. Oh, they don't even go there.
There are many definitions of species. A common
right.
definition is a group of organisms capable of interbreeding and producing fertile offspring.
Right.
And separating from other such groups. So for viruses...
they breed true, obviously.
Poliovirus type 1 can all... But, poliovirus type 1 and 2 don't They do recombine, actually.
Okay.
So they were called polio type 1 and type 2 for other reasons, not for species reasons. So all right, I don't really like species for viruses.
No.
Family and genus is good for classifying.
All right.
So how did we do this? Well, years ago, before we had any molecular biology, we did classification of viruses, and we classified them on a number of characteristics other than what we do today. We used to do the nature of the nucleic acid,
Right.
the symmetry of the shell,
Okay.
capsid, enveloped, etc.,
Yep.
whether or not it had a lipid membrane, and the dimensions. So various sizes, or if you're beyond a certain size, you're usually not in the same family, basically.
What about host characteristics of infection?
Host characteristics, disease, tropism, absolutely.
Yeah, sure.
Antigenic character.
Right.
But nowadays, it's mainly done by sequence. Because it turns out that the sequence really tells you everything. And you can put viruses in families and genera just by the sequence.
Well, Vince, let's go back to one of our earlier sessions where I was asked to give a little description of West Nile virus as an example of a single virus species, right? The West Nile virus.
Mm-hmm.
There are 23 known isolates, all different. They differ by their genomes.
Isolates.
Right. So,
now we have hundreds of swine flu.
of course.
H1N1 2009
So
isolates.
what would you call you'd call them isolates, of course.
They're isolates, absolutely.
Subspecies.
No, not even subspecies. They're
Right.
just isolates because it's the same species and you're just getting different isolates.
But they're geographically restricted, apparently. So that's how come we knew that the West Nile virus that we're dealing with in the United States came from the Middle East and not from, let's say, the south of France. They stay put.
Well, that's not always a fault of the virus.
Oh, no,
right?
no, I wasn't saying it at all. It might be the fault of the mosquito or the host,
Sure.
the mammalian host or something else, right?
The weather, who knows?
But isn't it interesting, though, because this virus originated in the West Nile district of Uganda.
Right.
And it spread throughout Europe and Asia. And even in this country now, it's become different by natural selection.
Well, that makes sense, right?
It does to me.
Because everything is different here than it is in the
So
West
the
Nile
host
district.
influences,
Of course.
not just the virus structure, but the virulence as well, because the virulence in this country has gone down over the initial input.
Are you sure?
The death rate from West Nile virus in the beginning was over 8%, and it's now below 2%. I know what you're going to say.
Is some immunity that's protective?
Or because we recognize it earlier and we treat them sooner, it's possible for that too. But
have experiments been done where the effect of the different isolate sequences on virulence has been studied? Probably not.
Oh, no. I think it has been studied. In fact, this guy Vincent Dick in the Pasteur Institute in France has done those studies because he's got all 23 isolates, now 24 from the United States. That's the hope, that this virus will attenuate as it's serially transferred from host to host, so that the crows won't all die off from this thing eventually.
Okay, what does the virus care about crows?
Well,
what do crows have to do with people? And what does this have to do with classification?
I think it has something to do with understanding how viruses change over time, because what you're suggesting is, from this classification, at least at some point, there's no change, that is, in its general shape, its general characteristics, but there's always change going on, as we've talked before about influenza virus, for instance. There's always change going on, right? So it still maintains its general characteristics.
Sure.
But and yet it remains...
Dick, a DNA virus remains a DNA virus.
Right.
You have a capsid, you can always have a capsid.
That's exactly
You're
right.
not going to acquire an envelope.
But the sequence of DNA changes.
The sequence changes.
Right.
And how much that's expressed into protein depends on the flexibility of the virus and its structure and its proteins and so forth. So flu happens to be very plastic. Its glycoproteins can handle an enormous amount of amino acid changes. Other viruses are much less. Measles makes a lot of errors in its RNA genome, but those are not found in the proteins because they're lethal. Measles is not as plastic as influenza.
I would presume
we don't
rabies
understand
is,
why.
Not so plastic either.
Yeah, it doesn't vary all that much.
Yeah.
So this classification now, you can look at the sequence and say, this is a poliovirus, no doubt about it.
Right.
And interestingly, so let me give you an example of a classification.
Please.
The family Picornaviridae or the picornavirus family. A genus is the enterovirus, and one of the species is poliovirus.
Okay.
Another species is Coxsackie virus.
Okay.
Polioviruses and Coxsackie viruses, if you co-infect cells, will not recombine.
What about rotavirus?
However,
is that in there too?
However, there are some examples of naturally occurring recombinants which seem to have sequence from polio and Coxsackie, and you can create such recombinants in the laboratory.
Wow.
So the classic definition of species is not really applicable. Another genus within the Picornaviridae is rhinovirus.
Mm-hmm.
Now, the organization that handles classification of viruses is the International Committee on the Taxonomy of Viruses, the ICTV. And every year they have a meeting and they review all the new proposals. The new proposal is that the enterovirus and rhinovirus genera are going to be combined because sequence analysis shows they should be the same genus.
Vince,
What did you say about rotavirus? It's a different family.
just asked you if it was in there or not.
It's a
'cause
rheovirus family.
But it's an
Rheoviridae.
enterovirus too, though.
It is an enteric virus, but, and you know what? It's also icosahedral. But it has very different characteristics.
Okay.
It has 10 to 12 pieces of double-stranded RNA. It has a double-shelled capsid.
Wow.
It's much bigger.
Wow.
Totally different.
Okay.
So that's why it's in a different family.
Okay. Now, $64,000 million question. As of right now, at this very moment, as a human being, how many viruses will infect me of all the ones you know about? How many different viruses infect humans?
Well, the first question is how many different viruses do we know about?
Okay, that would be my first question. How many? And you're talking about animal viruses now, correct? Yes, because plant viruses are over here someplace.
No, we can.
You want to throw in
we
plant
could-
viruses also?
In terms of the total number of
Viruses ever
viruses?
discovered.
yeah, let's see. I got that number here.
Oh, it's big. It's a
Okay.
big number.
So far we have 40,000 virus isolates. This is as of 2008 from bacteria, plants, and animals in 73 families, 287 genera, and 1,950 species.
Wow.
But remember,
How many altogether? Only 1,550?
1,950
950.
species. Identified, though, isolated.
Sure.
Remember, there are a million virions per mil of seawater, most of them unidentified.
Exactly. That's what I was going to say.
How many infect you? I don't know the number. We could count it, but I would say no more than a few hundred known viruses. You know, we got the picornas, the paramyxos, the influenzas, the rhabdos, the herpes, the pox viruses, and I'm missing many more, polyomas. But I'm sure there are many more that we haven't discovered.
Right. Yeah, they emerge regularly from the wildlife that we keep disturbing.
So the new arenavirus from Africa that we talked about last time just emerged. Never seen it before, but it was
Exactly.
there. I can guarantee you it's been
Absolutely.
in some animal in the forest of Africa for some time,
Sure.
and there will be many more. It's all a matter of what we look for. This is the age of virus discovery, as you know. We have great techniques for detecting and sequencing new viruses, and if you look, you will find.
In fact, there was a TED lecture on that I was at.
Joe DeRisi,
That's right.
the virus, and of course our colleague here, Ian Lipkin, you know, is also a virus pirate. Is that the right word, or a virus?
Virus hunter.
Virus hunter. And they are training the next generation, of course. In fact, out of Joe DeRisi's lab is David Wang, who's at Washington University in St. Louis, and he's a virus hunter.
Yep. How do you hunt for a virus, Vince?
You just sequence nowadays. It used to be that we did microarrays. You know what a
Right.
microarray is?
I do, of course.
Do our listeners know?
I think we should tell them.
You take a glass microscope slide and you spot with a robot hundreds and hundreds of small sequences from whatever you're looking for. So if you want, you could put a sequence of every virus on this chip. It's called a chip, but it's a glass slide.
Yep.
And then you can incubate that with a sample, hybridize.
Yep.
But we don't do that anymore.
What's the reporter molecule like? I mean, how does that actually work?
So what you would do is, let's say I take a little snot from you and I extract RNA because I want to find a virus. I would then turn that RNA into DNA,
Okay.
and the nucleotides I use would be labeled with a fluorescent dye, either a green one or a red dye,
Okay.
and that would be hybridized with the with this microarray chip, and then it's read by a fluorescent reader.
Got it.
This is a beautiful technology, but you're limited by the sequences that you put on the chip, right?
Of course.
So you're limited to known sequences.
Right.
Now what we just do is take that snot, extract the RNA, convert it to DNA, and sequence it.
Easier to do it.
Because there's a new technique called pyrosequencing.
Pyro implies like you're going to burn something.
It does, but it has to do with pyrophosphate, not
Oh,
pyro
okay.
in the burning sense, because of the chemistry that's involved involves
Okay,
pyrophosphate.
fine.
Started by a company called 454, which was up in Branston. What's the town in Connecticut? Branford, Connecticut.
Brantford.
I'll tell you the story of 454. The man who developed this technology, he wanted to sequence his daughter's genome. She had some genetic disease, and he wanted to know, so he thought of this sequencing technology, and then the company has since been bought by Roche. Now, they can sequence 600 million bases for about $5,000.
Wow.
So you give them a sample, and they'll give you 500,000 bases,
Wow.
500 million bases, $5,000. And the cost is coming down. As you know, it'll probably be $1,000.
That's
very
amazing.
soon. So I take your snot, convert it to DNA, and send it to 454 and sequence it. And you throw away. And now this is why we have bioinformaticians involved. You have to pull out all the
Sure.
host sequences that you're going to get and all the known genes and so forth. And you get 100 bases of some virus that's in you.
Got it.
And that's how you do it.
Hmm.
And universities now are purchasing these machines, so you don't
Okay.
have to send them to the company. And we have one here at Columbia.
Nice.
And that's how you discover viruses these days.
Got it.
So, Dick, I would say you and I go off, we'll trap some animals, get some RNA, and
Sure.
sequence it.
Okay.
And just get out your credit card, $5,000,
Not a problem. I just signed a contract with a book company. I can do this.
and then you get a new virus.
Right.
You can write a grant to study it.
I can name it after myself.
But you wouldn't do that.
I
Right?
would not. I would name it after you, Vince.
So there's
I
40,000
want the cure
isolates.
named after me.
There are. If you took all the viruses in the oceans and put them end to end, they would go 200 light years
You know
into
what? That
space.
sounds like a Dorothy Parker statement.
Doesn't it sound like a Carl Sagan? So anyway, we're going to discover many, many, many new viruses in the future. So that's taxonomy. Taxonomy nowadays is driven by sequencing.
Got it.
In the old days, it was driven by phenotype.
Mm.
Today, it is driven by sequencing.
Wow.
So you're right. This was a good second thing to talk about. You have any questions about classification of viruses, Dick?
No, now that we know that it's not a life form and we can't treat them as species, we can just treat them as entities that we can identify by sequence. The other question, the big question I would have at this point then is, how different does a virus isolate have to be considered unique?
You want a number?
If you have one. How many base pairs different does it have to be? What is the, in other words, what I'm really asking is, what is the error rate for sequencing versus the actual difference between one isolate and another?
Yeah, well, this is a problem because the sequencing error rate is quite low, but the amplification,
there.
when you take RNA
Aha!
and you amplify it by polymerase chain reaction,
Right.
there you have error.
Yep.
And so that makes it difficult to look at low-level
You
variation.
should do repeats then, yes.
Well, then you're going to get errors somewhere else, and then
True.
you don't know which one is real.
So how many of the real sequences that we have are actual real sequences?
See, Dick, the problem is they're all consensus. They're all sequences of the majority population.
Right.
So, viruses are quasi-species.
Yeah.
We haven't talked about this, but it's not one sequence. When you isolate a virus, it's not just one sequence. It's a mixture. Every
course,
virus
of course.
particle has a different sequence.
Right.
But when you sequence the population, you get a consensus.
Got it.
And so it doesn't reflect the diversity of the population. Now, with these deep sequencing techniques, as they're known as, we have the potential to take a virus stock and sequence and learn all the different variants in that stock.
Got it.
But you're going to be limited by the error rate
Right.
of the amplification step.
Yeah.
And
Which
there
is
are probably
what?
ways. Oh, the most faithful polymerase is. I'm going to make. I don't want to make a statement here. Let's look it up.
No, he doesn't want to get a letter like I get. I get all the letters. He doesn't get any of them. That's not
I
fair.
don't mind. I don't mind. So the most error-prone PCR thermal enzymes that are used in PCR make an error about every 1,000 bases.
Every 1,000 bases.
But there are high-fidelity polymerases that have much less error rate.
So wouldn't you have to go back after you amplified and got your virus isolate and we're pretty sure of what it was? You could go back with the polymerase that doesn't make so many errors and get the actual sequence.
Well, if you're just sequencing a viral genome, you can do that. But if you want to know the variation in a stock, it's harder.
Ah, right.
Here, let's see what the error rate is. So there are high fidelity enzymes that you can use.
So do we really know Craig Venter's sequence, or are we just looking at an approximation?
Oh, well, they do what's called, each position is done many times. It's called the coverage.
Okay.
So for a genome, and like us, human or
Which shouldn't vary,
genome,
no
they
matter where.
want 50
That's
to
right.
60 times coverage at each position. In other words, you've sequenced each base 50 or 60 times.
Wow.
So a mammoth genome sequence was recently published. I don't know if you saw that.
Yes, I did actually. It was very interesting.
And they had their DNA was very degraded.
Yeah.
It's old. It was 20,000 years old.
Yeah, old.
But they got six-fold coverage at each position, but they want to get 60
Right.
to have more confidence.
Right.
And the other problem with that sequencing project was that the runs were very short, one or 200 bases. So it's hard to
stitch it together.
together.
Sure.
So they used the elephant genome as a template to align it. It turns out that the mammoth genome was only 2% different from elephants.
That's amazing.
So there was not much pressure going from mammoths to elephants, whereas going from chimps to humans was more, it's like 5%.
Yep.
We had to get up on 2 legs to escape the tigers and lions that
That's true.
wanted to eat the chimps, right?
Yep.
That was the selection pressure. All right, PCR error rate. We have to get an answer because I will not
Sleep tonight until he knows.
I can't record this without an error. Okay, here we go. So the 1 in a million instead of 1 in 1,000.
Pretty good.
So there are different sources of these enzymes.
Right.
Pyrococcus furiosus.
Ooh.
And then there are enzymes.
Is that an
from,
extremophile?
Yeah, they're from the vents.
Okay.
Bottom of the ocean, very hot.
Okay.
And so these enzymes are put through thermal cycling, and so they have to be thermal resistant.
Sure.
So you go from about 1 in 1,000 to 1 in 100,000 to 1 in a million. Those are the good ones, 1 in a million errors. But in a 10 kb virus genome, it's not bad. But in eukaryotic big host genomes like ours, it can be a problem.
Right. So now we have the structure. We have
Classification.
So now we have to start talking about the process of infection.
Yeah.
This episode of TWiV is brought to you by our friends at Citrix, the makers of GoToMyPC, the premier program for remote access. If you ever feel chained to your office computer, you have to be at work, you have to survive traffic just to get in to work on it, why not work at home remotely? With GoToMyPC, you can access all of your office computer's files, even read your email, and reach network resources from anywhere in the world where you have an internet connection. And it's incredibly simple and incredibly secure. You go to gotomypc.com/podcast and you can sign up for a free 30-day trial. All you do is a few clicks of the mouse. The program is installed on your office computer. You get an access code, and then from any computer in the world, use a browser. Go to gotomypc.com, enter your access code, and presto, you have access to your computer's files, email, and resources. So remember, you can have a free 30-day trial. Go to gotomypc.com/podcast. That's gotomypc.com/podcast. We thank them for their support. Today, there are 2 emails that I want to use to discuss the swine flu immunizations of 1976. The first one is from Gus, and Gus writes, "Hello, Professor Racaniello. First, I want to say that I love your podcast. I'm fascinated by the topic and may have majored in virology if I was exposed to this material earlier in life. I'm a chemistry major and have worked in the semiconductor manufacturing field for more than 20 years, building integrated circuits, and now work for a small company in Monroe, Washington, making state-of-the-art compact, high-powered, solid-state laser systems. I'm glad you've decided to devote part of the show to basic virology, so-called Virology 101. Although I understand most of the material on the show, some of the common virology terms used by you and Dick are foreign to me. In future virology primers, it would be helpful to define and explain some of the more common virology terms commonly used in the field, which may not be so common to us lay people. For instance, I've heard the term negative strand RNA mentioned a few times on the podcast. It would be nice to explain exactly what a negative strand RNA virus is and how it works. I Googled the term, but the definition I found online is fuzzy. The negative strand virus was explained as the antisense strand of RNA, which does not encode for mRNA. mRNA encodes proteins. What does the antisense strand of RNA encode for, and how does it work? Okay, let's take that first before we move on to his last question. Negative strand is the opposite of the plus strand, and this is with reference to RNA. Plus strand RNA is the RNA that is used to encode protein. So in the cell, mRNA is positive polarity or plus strand, and that is the RNA that is translated on ribosomes to make proteins. And many RNA viruses have plus strand RNA genomes, like the picornaviruses, of which poliovirus and rhinoviruses are members. So plus strand RNA viruses have an RNA in their virion that can be directly translated when it enters the cell. The plus strand RNA is recognized by the cell and is translated. Now, as you know, there are many viruses that have negative strand RNA genomes, and influenza virus is an example of one of those. A negative strand is the opposite of a plus strand. It cannot be translated by the cell's translation apparatus. Okay, so in order to make protein from influenza virus genomes, the negative strand RNA first has to be copied to a positive strand. So the influenza virus RNA, which is negative stranded, enters the cell, is transported into the nucleus where it's copied to a plus strand RNA, and then that goes back out into the cytoplasm and is translated by ribosomes. So the plus strand copy of the influenza virus negative strand RNA can be translated. So this brings up an interesting issue. Viruses with plus strand RNA genomes don't need to carry any enzymes into the cell in order to copy their genomes, because their RNAs can be recognized directly by the translation apparatus of the cell. In contrast, viruses with negative strand genomes, like influenza virus, have to carry an enzyme into the cell which will copy the negative strands into plus strands. And that's because the cell doesn't have enzymes that can copy RNAs such as viral RNAs. The viral RNA of influenza virus, the negative strand RNA, cannot be copied by the cell into a plus strand, so the virus has to carry in an enzyme. So all negative strand RNA viruses have to carry in an enzyme, an RNA polymerase, to copy their negative strands RNAs into plus strands that can be translated into protein. Plus strand RNAs don't have to do that. So I hope that's clear. We'll certainly go over this in the segment of Virology 101, which deals with replication of genomes of viruses. But it is an important concept. I understand we've mentioned it a lot. So that's what it means. Negative strand, the opposite of plus strand, cannot be translated. All right, so Gus's second part. Here's one more last item. I will post a link to a suppressed 60-minute episode back from 1977 to 1978, which covered a swine flu vaccination campaign that seemed to go wrong. I'd really like to hear your perspective on this 60-minute episode. So he gave us a link to an episode of 60 Minutes, which explored the adverse effects associated with the swine flu campaign. Thanks much. P.S. Does Dick have a Twitter account? I follow both of you and Alan on Twitter, but haven't been able to find Dick's account, if he has one. No, I'm sorry, Dick doesn't have a Twitter account. He simply is not interested in doing all of these online social media type things. So the only way you can contact him is by emailing twiv@twiv.tv. Okay, now Swiss Compass wrote, regarding the probably impending swine flu vaccinations and concerns over Guillain-Barré syndrome or similar fears, could you please consider discussing this issue a bit more deeply? There is a growing resistance to accepting vaccinations and a lot of confusion on blogs and websites. A recent argument against accepting vaccinations is a 60-minute report from 1977 to 78 on swine flu, And he sent a YouTube link to the same video that Gus had sent, the 60-minute episode from 1977 on swine flu and the adverse effects. Please consider discussing this issue on TWiV and possibly laying out simple facts that can be used to dispel the fear, uncertainty, and doubt. Also, is the National Vaccine Information Center a credible organization? The National Vaccine Information Center is a organization which is aimed at allowing the public to make their own decisions about vaccinations. As far as I can tell, there are no virologists associated with it, so I don't know how they can present a balanced view about viral vaccines. So I don't want to make a statement about whether it's credible or not, but in my view, it doesn't really present all the information that you need to make a decision. What about swine flu immunizations and Guillain-Barré? So let's go back to 1976 and the outbreak of swine flu at Fort Dix, which is a military fort in New Jersey. So in January of 1976, there was an outbreak of febrile respiratory disease. There are 19,000 people at Fort Dix at the time, and virological studies showed that it was a swine influenza virus, and it was called A New Jersey 76 HSW1N1. So the hemagglutinin at the time was called HSW1 because it was a direct descendant of the hemagglutinin of the influenza virus that infected pigs starting in 1918. Now at Fort Dix in that year, in January 76, the virus appeared to infect about 230 soldiers, caused severe disease in 13, and one recruit died. Now at the time, we thought that the 1918 pandemic of influenza was caused by a swine virus. We didn't have the virus that we have today, the rescued 1918 virus, which shows that it's most likely an avian virus. So we thought it was a swine virus, and the isolation of swine influenza virus from recruits in 1976 scared many people. So many scientists were concerned that the virus of 1918 had come back to Fort Dix and would soon cause a catastrophic outbreak. Now, in 1976, I was a PhD student at Mount Sinai School of Medicine in New York, and in the Department of Microbiology, and the chair of that department was Dr. Edwin Kilburn, who was a very well-known influenza researcher. And he, in fact, was responsible every year for producing the high-yielding influenza recombinants, which were used to produce influenza vaccines. Now, he and others felt that it was important to immunize the population against this virus, so they convinced basically the U.S. Public Health Service to contract for producing 150 million doses of vaccine against the HSW1N1 virus. In March of that year, President Ford announced a program to inoculate everyone in the U.S. against swine flu. And those of us who remember, he went on television and got the shot on TV. In October, the immunizations began. The whole program was stopped in December. Only about 45 million doses were distributed by that time because it was clear that the virus never went outside of Fort Dix. In retrospect, we probably should have stockpiled the vaccine and waited to see whether it would spread. Now remember, first outbreak was January. The flu season is over at the end of May, so we don't see flu typically over the summer. So the idea was that it would come back in the fall. That's why we started immunizing in October. But in fact, it should have spread. If it was going to be a pandemic strain, it should have started spreading in January and February, but it did not. There was no evidence for spread outside of Fort Dix. Now, why? Well, the fort is quite isolated, and there's limited contact between basic trainees and others who travel outside the facility. And the older people at the facility may have been immune because there were some vaccines given to military personnel from 1955 to 1969 that had a swine influenza component to them. The other issue is most likely this wasn't the right virus to circulate or be transmitted among people. It was probably a accidental infection from a pig, and there was limited circulation because, in contrast to the 2009 swine origin H1N1 virus, it just didn't have the right collection of genes to allow it to spread efficiently in people. Today, of course, we know that the 1918 influenza virus is probably derived from an avian virus, not a swine strain. And if we knew that in 1976, we never would have carried out this immunization campaign. Now, getting back to how this virus got into Fort Dix, we really don't know. We don't know the origin of A New Jersey 76. Of course, one theory is that the swine virus was brought to the fort early in 1976. In January, the recruits were returning from the holidays, so some of them might have had contact with pigs and brought the virus in January, but none of them who were interviewed admitted to having any contact with pigs. So we really don't know the origin of that virus. And it circulated for about a month at Fort Dix and then disappeared, and that was the end of it. So really, in retrospect, we should not have started this immunization campaign. But of course, hindsight is always 20/20. We've certainly learned a lot from that experience. Now, one of the unfortunate consequences of the 45 million people who got the vaccine, there were about 500 cases of a syndrome known as Guillain-Barré, Guillain-Barré. Now, this is basically an autoimmune disease. The body's immune system attacks the peripheral nervous system. And we haven't talked much about autoimmune diseases on TWiV, but basically you make immune responses to foreign antigens, but sometimes you make immune responses against yourself, and that's problematic, and you have disease. And we'll probably talk about that in one of our Virology 101 episodes dealing with pathogenesis. Now, the symptoms of this disorder are varied. They include limb weakness, tingling sensations in the legs. So the illness begins in the legs. You can have a partial paralysis, and that can move up the body to the trunk and eventually to the arms and upper body. When it does reach the upper body, it's considered life-threatening because the respiratory muscles can be compromised and you have to be put on a respirator. Most people recover from Guillain-Barré, but about 5 to 6% of them may die. So there were a few deaths associated with the Guillain-Barré cases, the 500, which occurred again after temporally associated with the swine flu immunization of 1976, the fall of 1976. So Guillain-Barré is a syndrome because we don't know what causes it. We don't have a specific disease-causing agent. It's a collection of symptoms, so we call that a syndrome. It has been associated not only with the 1976 swine flu vaccination program, but with other infectious diseases and with bacteria in particular. A bacteria called Campylobacter jejuni is another common infection that triggers Guillain-Barré syndrome. And there are other respiratory and intestinal illnesses that are associated with Guillain-Barré. The 1976 swine flu vaccine is really the only influenza vaccine that has had significant association with Guillain-Barré syndrome. We don't know why this is the case. A number of theories have been set forth as to why, and one of them was that the vaccine may have been contaminated with this bacteria, Campylobacter, but that's not true. That's been disproven. Another was that the vaccine somehow was contaminated with material that induced antibodies against neurological tissues, but that hypothesis has not been supported by any experimental results. So we really don't know why that particular vaccine induced Guillain-Barré syndrome. So remember, it was given to 45 million people, and each year more people than that are given seasonal influenza vaccines, and there is a lower association with Guillain-Barré in those. So the real question is whether the vaccine against the 2009 H1N1 swine origin influenza virus, the current pandemic strain, is going to be associated with Guillain-Barré. And of course, there's no way to predict this at all. Remember, these vaccines are produced by making a recombinant or a reassortant between the strain, in this case the 2009 H1N1, and a high-yielding virus which grows well in eggs. And the reassortant will have the surface glycoproteins, the hemagglutinin, and the neuraminidase of the 2009 H1N1 virus, and other genes from this high-yielding strain. Now, the 1976 vaccine also was an H1N1, but it was quite different. And most of the other proteins of the virus were derived from the high-yielding virus. So the only difference would be the surface glycoproteins between the 1976 and the 2009 H1N1 vaccine. So I see no reason why the hemagglutinin or the neuraminidase would induce Guillain-Barré on their own. It could be a contaminant, but as I said, that's never been proven. And the hemagglutinin and neuraminidase of the 2009 virus are quite different from that of 1976. So, in the end, you have to balance the side effects or the potential side effects of a vaccine with the seriousness of the disease. We still don't have a good handle on the precise mortality or the case fatality ratio of this new strain. That's not something we'll have for a while because we have to do some statistics to figure that out. The current numbers look like it's about 0.4%, which is certainly higher than the incidence of Guillain-Barré. But I will be getting the swine origin vaccine in the fall of 2009. I will take my chance because I don't want to get influenza. And I see no reason to think that this vaccine will be associated with Guillain-Barré to the extent that the 1976 vaccine was. Now, the 60-minute episode, if you have a look at it, that was submitted to us by the two listeners, deals with the whole 1976 controversy. First, the issue of why we were immunized at all, because it didn't look like this virus was spreading. And of course, if you compare the spread of the 2009 virus with that of the 1976 swine origin virus, they're quite different. And it was quite clear very quickly after the first isolates of the 2009 virus that it was going to spread globally. And we had no such indication in 1976 for many months. So again, in retrospect, it was clear that the virus was going nowhere. So we probably shouldn't have used it, but it was approved by scientists, by virologists, by the Public Health Service, and that's a decision we have to live with. And also the 60-minute episodes focuses on the unfortunate aspect, of course, and that is that a number of people contracted Guillain-Barré, and some of them had permanent paralysis. So we went from being healthy to being disabled as a consequence of receiving that vaccine, which in the end wasn't necessary at all. So that's extremely unfortunate. But I see no reason why the same events will unfold with the current vaccine. I see no scientific reason to believe that the new vaccine will have similar effects. So that's all I can tell you about the 1976 swine immunization campaign and Guillain-Barré. I'll post a few links to articles in the show notes. And if you have questions, of course, do send them to TWiV at twiv.tv, and we'll get to them as soon as we can. I thought you would like to hear that sooner rather than later, so that's why I've made this little discussion on my own. I do want to close with some science picks of the week, and you may have heard Dick mentioning a book earlier called The Five Kingdoms, an illustrated guide to the phyla of life on Earth. And that's by Lynn Margulis and Carlene Schwartz. And it's basically a review of all the phyla from bacteria all the way up to higher organisms. So if our discussion of the kingdoms and phyla confused you, you might want to have a look at that. It's apparently a very, very good book. My pick is a book called A Genetic Switch by Mark Tashney, and this was a 1986 book from a virologist who worked on bacterial viruses and used them, in particular bacteriophage lambda, to understand how genes are regulated. It's a classic. It's very well written, very clearly written, and I think you'll enjoy it. A little paperback book that's very accessible. And of course, if you're interested in other good science podcasts, check out sciencepodcasters.org and promednetwork.com, where you can find other great science podcasts. Remember that TWiV has a website where we post show notes, twiv.tv. Do send us your questions as usual. Tell us what you think, what you'd like to know, what we got wrong. twiv@twiv.tv. You can also send us an MP3 file if you want, or you can call us on Skype. Our username is TWiV Podcast, all one word, and you have about 10 minutes to leave a voicemail. You've been listening to This Week in Virology, the podcast all about viruses. Thanks for listening. We'll be back next week. Another TWiV is viral.